then you can just proceed. Davis, you can confirm that you can hear me?
Oh,
all right.
sorry, I had muted. I can get you very clearly.
All right. So maybe you can just move closer to your audio device and then you can just proceed.
Okay, thank you. Good afternoon, everyone. Good afternoon, colleagues, ladies and gentlemen. Hope I'm audible enough as we begin this presentation today. I've been introduced by my colleague, my professional colleague, Mr. Obade. There's nothing more on that. And I'm just going to go direct to the presentation for the day. So our topic just a bit. I'm having a frozen screen as I try to navigate. Okay, thank you. Our topic today is ensuring or enhancing laboratory reliability through aligning fit for purpose and fit for use. I'm very sure we as medical laboratory personnel, we as professionals in this field, statements of the kind of this nature as to purpose, fit for purpose or fit for use are not new to us. And that's why I want to take this opportunity to take us through this presentation to build up a basis for discussion so that we go step by step on what we intend to have tackled today. This topic actually touches on all of us. It cuts across, it cuts from a class B lab to all the way to a class G lab. It cuts away from non-accredited labs to accredited labs. So it is actually a topic for everyone. And I want to welcome you to today's topic. So what is the basis? When we hear fit for purpose, fit for use, why do we come up with such a topic? Or what is the basis behind such a topic? And we shall draw our basis today from the ISO 15/2022 requirements. These can also be found in the general GCLP requirements for our labs. So where do we get the basis for this topic? We go to ISO 15/2022 alignment and a specific approach for fit for purpose. We draw our inference to clause 7.3.1, and that clause actually talks about selection, verification, and validation of methods. And it says, laboratories of any kind must ensure examination processes are appropriate for the intended use. Okay? Clause 7.3.2, which actually talks or addresses the fact to validation of laboratory developed or modified methods, requires full validation to demonstrate that the methods are actually fit for its intended purpose before use on patients. On clause 6.4, with regard to equipment, the clause points out that equipment must be selected based on ability to meet analytical and operational requirements of the lab. So That actually demonstrates why this topic of the day is very, very important, whether accredited or not accredited. Just bear with me, colleagues. I think I'm having transmission issues. Okay, thank you. Clause 7.3.1.3, which talks about verification for performance. It tells us that even for commercial methods, labs must verify that the method performs as expected in their specific operational settings. Clause 6.2, which talks about personnel competence. It addresses the fact that staff must be trained and assessed to ensure that they can consistently apply the methods correctly. Clause 8.5, that talks about continual improvement. It encourages monitoring of real-world performance and making adjustments to ensure fitness for use. Clause 7.7, post-examination processes, ensures that results are reviewed, results are interpreted, results are reported correctly, confirming that the entire process is functioning as intended. We are just having an overview of what actually does the ISO 15189-2022 requirements dictate with regard to purpose for use and fitness for use. Having highlighted the basis for this discussion, the basis for this topic, we then draw our attention to what are the objectives? What are our intentions? What information do we intend to bring to you? What do we intend to achieve at the end of this training? Number one, by the end of this training, we hope to have achieved or given a proper definition that befits your utilization, a proper definition for fit for purpose, and a definition for fit for use in lab operations. Number two, we hope to promote alignment between technical requirements and practical usability. Number three, we hope to identify risks of misalignment in procedures, equipment, and workflows. These are actually risks that may arise from misalignment between fit for purpose and fit for use. And finally, we hope to achieve an enhancement in decision-making and operational efficiency across the lab. This is just a hypothetical illustration to help us have a feeling of what we are going to discuss. On my left, I have a syringe. This syringe has been produced by manufacturers. The manufacturers of this syringe, before deploying it to the market for circulation, they indicated that in their controlled laboratory setting, the syringe performs as per requirements. So it has met the concept of fit for purpose. But then it is our responsibility as people, as the end users of this product, to ensure that the product is fit for use in our settings. And how do we do that? We have to ensure probably that the packaging of the needle or the syringe is still intact. We have to evaluate that there's no contamination of this syringe. We have to make sure that after usage, there's proper disposal. If these concepts are actually not factored in the usage of this syringe, it means, yes, it's fit for purpose, but from the end user perspective, it's not fit for use. On my right, we have a pipette. This pipette comes from the market or from the supplier with a certificate of analysis or a certificate of conformity, indicating that, yes, we have calibrated this pipette for accuracy, and it's very suitable for your biochemical assays. But then it's your responsibility as the end user to make sure that this pipette is fit for purpose. How do we make sure that this pipette is fit for purpose? We have to maintain it well. We have to train people who are operating this thing. They have to understand the processes. Is it forward pipetting or is it reverse pipetting? Okay? So at the end of the day, whatever that has been produced or put forth by the supplier or by the manufacturer has to be confirmed, and then other additive aspects have to be put in place to make sure that what is actually deemed as fit for purpose can actually be translated to fit for use. So what does fit for purpose mean? Fit for purpose in this context looks at, is our method, is our assay, is our process or tool suitable for its intended application or use in the lab? This actually is looked at in the context of relevance, contextual relevance. The method must generate data that is meaningful and sufficient for specific decision-making scenarios. Okay? And fit for purpose, for something to be fit for purpose, it must meet technical specifications. Technical specifications we are talking of, what is the accuracy? What is the precision? What is the sensitivity? What is the specificity? And these technical specifications are actually very essential to ensure that a method actually performs reliably or supports the intended clinical or diagnostic use. Examples for clarity. For example, if you want to diagnose malaria, you want a kit, a test kit that diagnoses malaria, that can actually pick up malaria antigens or otherwise. If you want to diagnose malaria, the kit or the test that you are using should be fit for purpose. And in this context, what does it mean? The method should be able to detect malaria accurately and reliably in the specific patient population and sample type. As I had earlier mentioned, this kit or this test methodology must meet technical specifications. And when we talk of technical specifications, we are talking about the sensitivity, specificity, reproducibility, among other aspects that we have previously discussed. Okay? So, this test does not need to detect every parasite globally. It only needs to be specific to what you need to get. And in this context is the malaria parasites. A second example, we are looking at HIV screening in blood donors. This means that the test that we need to employ in this context should be very sensitive, because these are blood donors. The test methodology must be very sensitive to pick up even the slightest or the earliest of the infections, okay? So for a kit to meet the purpose, it means its sensitivity has to be high. Whatever that the manufacturer has indicated has to be of the highest aspects, and it must be collaborated in the lab settings. We shall be looking at that. A test that is put out to the market to actually monitor glucose in diabetic patients must be accurate within certain range that affects treatment decisions. So whatever that has been put forth as fit for purpose in this context must actually meet the intended use in the market or in the specific lab settings. So now that we know what fit for purpose is, how do we ensure that whatever we are bringing in is actually fit for purpose? Because we cannot just rely on what the manufacturer is telling us. The manufacturer will say A, B, C, D, E, F, but we internally, as the end users of this product, we must have our own standards. We must have our own checklist that even if I want a kit for screening, a screening kit, it must have a BCD specification. If it's sensitivity, it has to be above 80 or above 90. That is an internal decision that has to be made. And it's you as the end users who actually decide on what to adopt, and what has to be adopted might actually be fulfilling to the requirements as well. So what is the checklist that can actually be used to give us or to ensure that something is fit for purpose? Number one, we are looking at kits that have been approved, that have actually received the necessary approvals. And when you look at necessary approvals, you can also, of course, decide to look at, is the kit FDA approved? Is the methodology FDA approved? Is the analyzer FDA approved? Okay. Does it meet ISO certification standards? Those are some of the questions that you ask yourself. Does it comply with the local health authorities? And we are talking, when we talk of local health authorities, like in Kenya, we are referring to an example as PPB. Does it meet the requirements of PPB? Has it passed? Okay. Then we look at manufacturer specifications. What is our intended use? Are we just bringing in a kit for screening purposes, or we want it to use that as a diagnostic kit? Because that gives you different aspects that you need to bring in for. For example, if it's for screening tests, you will actually go for a kit, a methodology with the highest sensitivity. Okay, but if it's for diagnostic purposes, you'll go for a kit, yes, as long as, in as much as sensitivity is important, we also go to for a kit that is highly specific to what we are looking for. So with regard to manufacturer specifications, we are looking at what is the intended use. Does the kit in the market actually match what you want to use it for? Okay. Then you look at the sensitivity, specificity, or accuracy or precision of these kits. Of course, we are basing on what the manufacturer is putting forth, because at the end of the day, we shall have to verify. So what is the shelf life, the valid shelf life and proper storage conditions of these kits? Can we actually meet them? Can we meet the requirements? You know, there's no need why you should bring in something that requires negative 70 freezer when you cannot achieve that. Okay, so we are just trying to, you internally, what are your requirements? What do you need? Okay, does what is in the market actually fulfill the requirements that you have at the back of your mind? Then number 3, on the checklist, we look at validation and verification. Have this kit or methodology been tested against non-standards? And when we talk of this, there's a concept, of course, it brings out a very big point of discussion, but there's a point or a topic on its own called metrological traceability. It actually comes in. Does it meet the standards? Has it been tested against non-standards? And did it pass? Does it have traceability? Okay. So does your method or does your test kit give consistent results across samples? Okay. That's what are there proper calibration procedures in place for this kit or methodology as such? Another aspect we need to look at is quality assurance, production quality assurance. Okay. Batch testing or QC documentation, is there relevant or accurate QC documentation aspects that have been put forth in this method? We are talking of certificate of analysis or certificate of conformity. Has the manufacturer actually given information, sufficient information, that this methodology or this analyzer or this test kit has been actually produced under strict quality assurance processes and can actually translate to reliable outcome. Then we also look at, is the supplier, of course, reliable with good support? We have people who can give you a kit, you adopt that kit and they disappear from the market, okay? They get you back to ground zero. So those are some of the things we need to review when you're looking at that. Then we look at number 5, usability and compatibility. Is it easy to use? Or is it easy to train staff to adopt whatever methodology that you're putting in place? There's no need why you should bring a very complex methodology in the lab that even training your staff is a problem, okay? The error rate goes very high, it shoots. Okay. Then you have to ask yourself, is it compatible with the existing lab system? For example, maybe I have a result transmission system. Is the analyzer I'm bringing in compatible with it? Those are the questions that I should ask myself. Are there clear instructions and support available? When we're adopting this methodology, do we bring in something that has no instruction? There's no support that comes with the analyzer or support that comes with the kits. In case of a problem, there's nobody to get in contact with. Then we also look at cost and availability. We are going for cost-effective aspects that are readily available. There's no need why you should go for a very expensive aspect that is not readily available in the market, okay? So reasonable cost for quality, and we're also looking at reliable supply chain. There's no way you should adopt something that is not. So if you have something, yes, it's very expensive, it's not readily available, it might be good in some areas, but it's not fit for purpose in your context, okay? So what are some of the questions that you ask yourself? How do you self-assess yourself? How do you self-assess yourself when you're looking at these things? Of course, I told you that fit for purpose actually looks at alignment, intended purpose, and contextual relevance. What is the relevance of this of this methodology or this kit? Does it meet your requirements? Does it actually meet the requirements to fulfill what you need to achieve at the end of the day? So with regard to that, number 1, these are some of the questions. It does not mean that these are the standard questions that you shall ask, but these are just some lead aspects that you can consider to adopt. You ask yourself this: what is the specific clinical or diagnostic decision this method supports? And in this case, probably you want to adopt a system that for screening purposes or a system for diagnostic purposes. You ask yourself that question. What is the specific clinical diagnostic decision that this method supports? Number 2, is the method aligned with the intended use? And number 3, what are the consequences if the method fails in the context? What is the consequence? For example, we put forth an example when we were having a discussion that if you want to screen HIV in blood donors, you need to get a very highly sensitive method that can actually be used to adopt this. So you ask yourself, what is the consequence if this screening methodology that we have put in place fails and is being used in blood donors? That is just a rhetorical question for our own consumption. Number 2, what are some of the questions that you can ask yourself to assess the technical performance? I said that when you are assessing whether the method is fit for purpose, you must have a predefined threshold. You internally, what do you want? What standards should be brought on board? So you ask yourself, with regard to accuracy, how close are results to the true value? With regards to precision, We are talking of consistency and repeatability of measurements. How consistent or how repeatable can these measurements be when generated from this machine or this testing kit? We talk of sensitivity. Can it detect low levels of the target analyte? Okay, specificity. Can it distinguish the target from similar substances? Those are some of the questions that we ask ourselves. With regard to validation and verification, you ask yourself, has the method been validated for the specific matrix or sample type? Were performance characteristics verified under the actual lab conditions? Okay. Are there documented results from method comparison studies? Because these are some of the aspects that you are going to pick them out. You are going to pick it out. Okay. Number 4, quality control and monitoring. What internal QC measures are in place to monitor ongoing performance? Does this methodology have internal IQC aspects that can actually be used to monitor performance as it comes? How frequently is the method reviewed or validated? Are there trends or deviations in QC data that suggest risks? With regard to regulatory and compliance, you ask yourself, does the method comply with the relevant regulatory standards? Is it FDA approved? Is it CAP approved? Are there documented SOP and training records for personnel using the method? Because these are very important questions that you should bring forth to bring out the aspect of fit for purpose. Risk mitigation. What controls are in place to reduce the risk of false positive or negatives? What controls in rapid kits, what controls are in place to ensure that the risk for false positive and is there a contingency plan if the method fails or produces unreliable data? Very important questions that we must ask ourselves. Then we transition to fit for use. What does fit for use actually bring forth? Fit for use means that even if a test is scientifically sound, even if it has been proven that it's fit for purpose, the method or system is ready and capable to perform reliably in real-world conditions. Even if a test is scientifically sound, the test methodology has been deemed fit for purpose. It is our responsibility to ensure that this method or system is actually ready and capable of performing in real-world conditions. A test methodology or test kits were manufactured in Europe. They were deployed for usage in Kenya, in Mombasa. Different conditions, different aspects of that. So the kit that performed well, or the methodology that performed well in Europe, the machine, the analyzer that was functioning properly in Europe, when brought in Kenya, does it function the same way as it was functioning the other end? So it is actually our responsibility to ensure, to evaluate that actually this machine is functioning effectively the way it was deemed during the laboratory manufacturing of it on. So this process of ensuring that actually the machine, the kits, or otherwise are performing well or reliably in real world is actually making what was deemed fit for purpose fit for use. Okay? So what was deemed fit for purpose must also work consistently in your lab environment. If it does not work consistently, It means, yes, it's fit for purpose, but it's not fit for use. And that cannot be adopted as such. When we are reviewing fit for use, of course, we look at how robust is your method to variations. How easy is it to use? How compliant is it to quality standards? Okay? So there is a statement down. While fit for purpose actually focuses on whether a method is suitable for intended applications, fit for use goes a step further to assess whether the method or tool is ready and reliable for actual use in practice, especially under real world conditions. So, in short, what does fit for use mean with regard to method, assay, or tools? When you say something is fit for use, it means this method, this assay, actually performs consistently under routine laboratory conditions. That's number one. Number two, it means this method, this assay, is actually validated and verified, not just theoretically, but practically. Number three, it means this method, this assay, or this tool is actually maintained, is actually calibrated, is actually controlled to ensure ongoing reliability. And finally, this method, this assay, or this tool is actually accessible and usable by trained personnel with the necessary infrastructure. So to summarize all this for better understanding, fit for purpose actually relies on manufacturer pre-qualification. What is the manufacturer telling us about this syringe? What is the manufacturer telling us about this analyzer? What is the manufacturer telling us about this pipette? What is the manufacturer telling us about this? Or what are the writers of this SOP actually telling us? Okay? But fit for use relates to method or procedure qualification in the real world. Somebody sat somewhere and decided to come up with a very effective procedure or SOP about gram staining. Gram staining, where it was tested and before its adoption, it was considered fit for purpose, okay? But then in your local lab settings, how do you ensure that, yes, this gram staining protocol that was declared fit for purpose is actually fit for use? You train your staff. You ensure that you have everything that is required in terms of requirements for gram staining. You do not bring in substitution. You make sure that you do competence assessment for your staff. You make sure that any competence failures leads to retraining as such. So that is how you declare something fit for use. So fit for purpose must actually be translated to fit for use for proper adoption and reliability of the outcome in the lab. So that is just important take-away points. Fit for purpose is manufacturer responsibility, or whoever, for example, if it's kits or analyzers or whatever tools that you are using in the lab. But when it comes to SOPs, that is the responsibility of the writers of that SOP. So, but when we talk about fit for use, is the laboratory personnel's responsibility. Okay? You must test the method, you must verify to make sure that it performs actually effectively in your actual lab environment. Of course, fit for purpose does not put into consideration real world variables, but fit for use considers real world variables like equipment, personnel, workflows, and environmental conditions. Different environmental conditions must be tested against what we are using to do that. So, what is the application? What is the application of fit for purpose and fit for use in our lab settings. How do we apply these concepts? And as usual, I like looking at concepts from the three phases of analysis. Number one, we look at the pre-analytical phase. How do we apply fit for purpose and fit for use concepts in pre-analytical phase? These are just some examples. There are so many aspects that you can actually consider. These are just some examples to make sure to bring out a better understanding of these concepts. Number one, sample collection tubes. The manufacturer produces specific sample vacutainers. And these vacutainers are actually validated before they come to the market, okay? For example, EDTA for hematology or heparin for chemistry, or no, for SST for chemistry or such. So since the tubes have been validated by the manufacturer for specific analytes, they are fit for purpose, okay, before they're introduced in the market. But then it is your responsibility as the lab to actually confirm that these tubes are being stored correctly as specified by the manufacturer, they're being used before expiry as specified by the manufacturer, and the staffs are actually trained and competent enough to select the right tube for the right test. That brings in the concept of fit for use, okay? Validation by the manufacturer, that is fit for purpose before they're introduced. But once these kits are introduced in the market, it's your responsibility as the lab personnel to make sure they are stored correctly, they are used before expiry, and the staffs are trained for usability. That is fit for purpose. With regard to transport media, of course, the manufacturer has specified that the media is designed to preserve sample integrity for specific organisms or analytes, okay? So it is our responsibility as the lab to verify cold chain is maintained during transport and the media is compatible with local workflows. So in that context, the lab actually applies rejection criteria consistently and documents reasons for rejected samples. With regard to patient preparation protocols, the writers of the SOP have clearly indicated that the patient must fast. The patient must be in ABCD posture or specific timing requirements based on sample type 4, okay? They have already declared that SOP fit for purpose. So how do you ensure as a lab staff, how do you ensure that this actually SOP is fit for use? The staff is tasked with the responsibility of ensuring patients are properly instructed and compliance is documented before sample collection. We have properly documented SOPs for sample rejection in our areas of work. This SOP actually clearly give a predefined criteria for rejecting hemolyzed, clotted, or otherwise samples. That is fit for purpose. It has been tested and it has been proven. So how do we translate fit for purpose to fit for use? The lab applies rejection criteria consistently and documents reasons for rejected samples. That is fit for use, okay? Those are just some of the examples, a few examples for us that we can use as building blocks in pre-analytical. In analytical phase, we talk of reagents and kits. Reagents have been validated for stability, specificity, and compatibility with the testing method. That is fit for purpose. The manufacturer has put forth, or the supplier has put forth that. So what does the lab do to actually ensure that these reagents and kits are fit for use? The lab checks reagent storage. It evaluates expiry dates. It evaluates consistency of results across batches. If all these things match, then the reagents and kits are actually deemed fit for purpose. Reference ranges. I know reference ranges is a whole discussion, but I'm not going into development of reference ranges. But then the manufacturer of a given system provides you reference ranges, okay? The manufacturer, for example, of a system, a testing methodology, or as an analyzer indicates that analyte AB for a normal population should be between 3 and 10, okay? Those are testing reference ranges where the production happened, on the population where the production happened. So these manufacturer-provided reference ranges based on population studies where the production happened, they are fit for purpose. But then you as a lab, you cannot adopt these reference ranges blindly, because what was fit for purpose in Europe does not actually translate to what is fit for use in Kenya. So what does the lab do? The lab verifies ranges, that these ranges are actually appropriate for local populations before adoption. And if they're not appropriate, you adjust based on internal validations, okay? Quality control materials. QC materials have been validated by the supplier or the manufacturer to actually ensure continuous monitoring of method performance. That is fit for purpose, as indicated by the supplier or the manufacturer. But how do we as the lab actually declare what the manufacturer has declared as fit for purpose? How do we declare it as fit for use? The lab runs daily QC, interprets results correctly, and takes corrective actions when needed, okay? Once these are done and they are okay, it means we have just validated, we have just verified the assertions of the manufacturer, and we have translated what has been deemed as fit for purpose to fit for use. Instrumentation. Equipments are actually designed and validated to perform specific tests as indicated there. That is fit for purpose. So the lab actually ensures these instruments are calibrated, maintained, and functioning correctly under routine conditions. And with that, the lab actually certified what was declared as fit for purpose is now fit for use. The method validated by manufacturer for specific analytes with non-performance specifications indicated there. It's fit for use. So we as the lab personnel, we verify that the method works with the local sample types, it works with the local instruments, and it actually meets performance criteria in practice. Once we do that, we are declaring what was declared as fit for purpose, we are translating it to fit for use. Post-analytical phase. We have come up with a very nice format. Somebody sat somewhere and came up with a very nice format on how to document your results. You have a very nice template, clearly and accurately presenting results. That is fit for purpose. How do we then translate what has been deemed fit for purpose to fit for use? The lab reports, we have to ensure that the lab reports are understandable, correctly formatted, and tailored to the clinical needs or clinician needs as such. Critical values alerts. We have a system actually that is designed to flag and alert critical results automatically. Okay? That is fit for purpose, because the entire purpose is to actually pick out what is critical and highlight it for you. But how do we translate this fit for purpose into fit for use? The lab confirms alerts are triggered correctly. Staff responds promptly to the alerts, and documentation for the alerts is very complete. Once we have done that, it means we have translated what was actually deemed as fit for purpose into what is actually fit for use. Data transmission systems. We have a laboratory information system validated to securely transmit results to clinicians. Yes, it is serving the purpose. It has been designed to actually serve the purpose that is intended to do. Yes, that is okay. That deems it fit for purpose. But then how do we ensure that what is fit for purpose is translated to what is fit for use? The lab ensures systems are integrated. The lab ensures these systems are functioning. The lab ensures that the data that is being transmitted does not have any errors. And the transmission is very timely. Once this is achieved, it means we have achieved fit for use. Results review protocols. We have very well-designed SOPs to ensure results are reviewed before release. That is good. It's fit for purpose. Okay? How do we ensure that then these SOPs are fit for use? The lab confirms results are reviewed by authorized personnel and discrepancies are resolved before reporting. Data archiving systems. The system, we have a properly designed system to store and retrieve historical data securely. Yes, it's fit for purpose. How do we ensure that it's translated to fit for use? The lab ensures data is backed up, accessible, and complies with retention policies and regulations. Now that we have had a discussion about fit for purpose, its applicability, its importance, its applicability, fit for use, its applicability, what is actually the importance? Why should we have this discussion in the first place? Number one, ensuring that something is fit for purpose and is fit for use actually secures or enhances patient safety. We can't run away from that because laboratory results directly influence clinical decisions. Okay. So once we have actually made sure that all the aspects of fit for purpose and fit for use have been addressed, it means whatever output that we are handing over is actually as reliable as possible. So if a method is fit for purpose or not fit for use, it can lead, if a method is not fit for purpose or is not fit for use, it can actually lead to misdiagnosis, delayed treatment, and inappropriate therapy. So the second importance is it supports regulatory and accreditation compliance. Of course, the basis that we started with, we were talking about what is the basis of fitness for purpose and fitness for use. It's actually inscribed in our ISO standard. Okay, it's actually inscribed in our good laboratory practices. So once we have this actually addressed effectively, it means we are effectively supporting regulatory and accreditation compliance. Whether you are accredited, yes, ISO compliance, but regulatory in terms of good clinical practice. So, as I had alluded, ISO 15189 requires labs to validate, verify all methods and processes. So demonstrating fitness ensures that the lab meets international standards and is ready for audits or inspections as such. Number 3, importance is it improves results accuracy and reliability. Fit for purpose ensures the method is scientifically sound. Fit for use ensures it works consistently in your lab. So together, they reduce variability and errors in test results. Number 4 importance is it reduces operational risks. Identifying gaps early prevents costly rework, sample rejection, or test failures. Number 5, it enhances confidence in our lab services. So of course, who are the end users of our results? They are the patients and the clinicians. So clinicians and patients actually rely on labs for trustworthy results, okay? So a lab that evaluates fitness builds credibility and confidence in its services, okay? Someone will ask a question, for example, why do you charge 500 shillings or 400 shillings for a H. pylori and somebody else charges 2,000, 3,000, 4,000, 5,000 for a H. pylori antigen test? And all this, you find that clients are not taking your 500 valued H. pylori kit, they are going for that is being charged at 5,000. The confidence, credibility, and trustworthiness of whatever outcome that you have been channeling out. Of course, fitness for purpose evaluation and fitness for use enables continuous improvement, is one of the aspects for continuous improvement. Ongoing evaluations for fitness helps identify areas for process optimization, staff training, and technology upgrades. Okay. Number 7, it actually supports resource optimization. This is achieved through ensuring that time, reagents, and equipment are used efficiently, okay? We are factoring in trying so much to translate what is fit for purpose into what is fit for use. It also prevents wastage due to repeated testing or invalid results. If we have all our systems actually declared as fit for use, it means the error rate is low. And once the error rate is low, aspects of repeated testing or invalid results are actually quenched in such. Okay? So colleagues, for us to achieve this, the take-home point is whatever specifications that the manufacturer has given, they must be translated into actionable outputs. The manufacturers or the writers of SOPs declare aspects fit for purpose. It's us as the end users, it's us as the people dealing with these aspects on a daily basis, practically, who declare aspects fitness for use. Okay? So whatever has been declared as fit for purpose must be declared as fit for use. Okay? What is declared as fit for purpose and not fit for use should not be adopted. The only aspect that should be adopted is what has actually been declared both fit for purpose and fit for use. Thank you very much. Thank you, colleagues. Back to you, Frederick.
Thank you so much for a very, very wonderful presentation. And truly, it was very, very encouraging and very interesting, very organized. And I can see that our participants have followed keenly. And at this point, we're just going to go into a few questions or comments from our members. And indeed, we are tuned in from across the country and without the borders. And to you, our presenter, there's so many comments here about the presentation, and maybe I can just read a few and then we can get into the Q&A section. So Duncan Perez says it's a good presentation. Joel Adanje is saying he's keenly following from Nairobi. And then I can see Irene Dairo is saying very interesting information. Martin Jogona says well organized and transformed into simple language for all. Very good work. That is Celestine Mulwa. And then Lawrence Maraka is saying very interesting now that I've known the difference in machines brought into the lab, whether they are fit for purpose and use. So I think the comments are a bit overwhelming. Evans is saying very informative and insightful. And to our participants, we want to say thank you for tuning in. And we know that for sure we need to keep on learning because learning has no end. And also we need to just know what you are doing. Peer-to-peer learning is very important. And also we just want to know best practices from where you are. And at this point, maybe you can just take a few questions and then we can proceed. So Davis, there's a question here, and I'll pick the first from Samuel Kipngetich. And I think this question keeps on recurring to majority of lab professionals. And he's asking, these two are confusing me. What is the difference between validation and verification? So Davis, you can take that. And then
thank you, Fred.
Take the second one, then you can just answer from Martin Jogona. Thank you, Martin, for the question. He's asking, having communicated to us this information, can personnel adapt their own analytical measuring range? So I think we can start with those two, Davis.
Thank you, Fred. What is the difference between validation and verification? Validation is actually what is put forth by the manufacturer. Okay? Those are the specifications, the technical specifications as highlighted by the manufacturer. So, in short, in relation to the discussion that we have had today, the validation as highlighted by the manufacturer translates to fitness for purpose. Okay? So when these analyzers or otherwise or kits come into market, it's now you as the end user to verify the assertions of the manufacturer. If the manufacturer actually indicated that in his validation report indicated that the specificity is 90%, it is your responsibility as the lab, the end user, to verify that indeed the specificity is 90%. So validation, in short, validation is manufacturer assertions, and that leads to fitness of purpose. Verification is from the end user to confirm the assertions of the manufacturer. And this actually, through verification, we achieve what we are calling fitness for use. I said when I was mentioning something about reference ranges. I told you a supplier or a manufacturer can actually give you reference ranges. But these reference ranges are based on population studies where the manufacturing actually happened, what was adopted. Let us say a machine is coming from Europe. There are high chances that population studies that were used to give these reference ranges actually exist in Europe, okay? And what works in Europe does not translate to what works to us in Kenya. That is why I say, once these reference ranges have been given, it is our responsibility as the end user to verify whether they conform with our local population. And if they don't conform, then it means we make adjustments based on our given local population. Thank you, Fred. So in terms of analytical adoption, yes, it can be adopted. Yes, you can create your own depending with the performance of what has been put forth. Thank you.
Thank you so much, Davis, for that. I'll move to the next. All right. I think these are just terms. I'll bring in this term. So explain, Victor Omoga is asking, explain sensitivity and specificity in relation to TrinScreen testing kit. So these terms sometimes we are not clear to many of us. So maybe you can get that. And then I'll ask the second one from Samuel Sigei. Thank you, Samuel, for the question. This presentation is really insightful. Thanks, Davis. How frequent are pipettes recommended for calibration? Thank you, Samuel, for that.
Thank you, Fred, for the questions. And thank you for the listeners for raising that. I'll not specifically pinpoint out TrinScreen, but I'll define specificity from the context of operationability. Sensitivity is the ability to pick out true positives, okay? Specificity is the ability to pick out true negatives, okay? So ideally, this is a broad-based kind of definition, but it cuts across, okay? It cuts across. But in the context, I believe I get what my colleague is trying to bring out is the sensitivity of TrinScreen. Can it only pick out HIV? Or can it pick out other things that might mimic HIV? And that's why, my brother, TrinScreen is a screening test, okay? We said that when we are actually designing, we need to look at what is the intention. TrinScreen is a screening test. So it has to have the highest sensitivity possible to pick out anything that mimics positivity has to be picked. But then there are follow-up specific tests now that can isolate what is HIV and what is not HIV. I believe, my brother, from that you have been answered. It is advisable to do yearly calibrations for pipettes. Yearly calibrations for pipettes is good as long as the handling or management of these pipettes is being done correctly. Follow the specific manufacturer specifications when it comes to management or handling of the pipettes. If those specific instructions are adopted too, it is actually suggested that you do an yearly reevaluation of calibration as such. Thank you.
Thank you so much. Maybe I'll get into the next couple of questions. This is from Wilfred Aria. He's asking, how does the concept of fit for use justify the reliance on RDTs in rural dispensaries lacking laboratory staff? Okay. Then I'll read the second one. How can a test method be considered fit for purpose, yet still fail to be fit for use in clinical practice? Thank you, Wilfred, for the questions.
Okay. Thank you, Wilfred, for raising that. In relation to RDT adoption, in clinical settings. It's not an aspect of fitness for use. It's an aspect of how do I put it? Convenience. Let me term it as convenience, because for you to declare an RDT fit for use, for you to declare it fit for use, one, you must have actually verified the specific technical aspects of this RDT. That's number one. Number two, you must do some rigorous training for the personnel that actually undertake this testing. Okay. Number four, there must be some technical competency training evaluation for these people, at least on a yearly basis, to see the adoption of that. Okay. Number five, are the errors documented and actioned? That is the question that arises. So the issue of adoption of RDT in remote areas is not actually an issue of fitness for use. It's an issue of convenience. Okay. There's an issue, a question that was raised with regard to fitness for purpose in. Fred, can you repeat the last question, please?
All right. Fred? So the question again.
Yes.
How does the, all right, I think, let me get the question. How can a test method be considered fit for purpose, yet still fail to be fit for use in clinical practice?
Okay. I said fitness for purpose is actually declared by the manufacturer. Okay. So it's actually your responsibility as the end user to either declare this instrument or this kit fit for use or not. Okay. We have had cases, although not so many documented cases, where manufacturers actually lie on certificate of conformance to give products on the market. So when this product actually reaches the market, the end user, when you verify the performance, it does not actually meet the specifications that you intended. For example, if internally you decided that for you to adopt a test kit, a rapid test kit, the sensitivity should be 80%, specificity should be 85 or otherwise, but then you get a kit in the market that the supplier or the manufacturer has indicated that yes, sensitivity is 95% and specificity is 98%. But when you do your own localized verifications of these assertions, you find that from the data that you have achieved, notwithstanding the prevalence of these conditions in the testing population, you find that probably your sensitivity is 75% of that kit and specificity is 70%. So one, it has not met your internal standards. Two, it has not met the assertions as declared by the manufacturer. So in short, yes, it was declared as fit for purpose by the manufacturer through omission or intent or otherwise, but in your own end final testing, it has failed to conform to that. So this method is, yes, fit for purpose as declared by the manufacturer, but not fit for use. Okay? That is why for established setting, before you start bringing in test kits, this is a take-home point I can try to give my colleagues. Kindly request the supplier to give you a sample of those kits for you to do verification first before you can buy from them. Okay? That is how it works. Send me a sample, I do verification on it. If it meets my specifications, I bring it in. If it does not, it was nice knowing you, and we will not adopt that. Thank you, Fred.
Okay, so I'll bring in two questions. One from, this should be Mabel Nduku. We have different companies supplying our lab with reagents. How can we be assured of quality and the end product is valid for our patient's outcome? And then Francis Mambili is asking, how do we ensure our equipment and test kits are fit for purpose and use? Thank you, Francis and Mabel.
So thank you. I think that is just one question in its context. So when I address it once, I address everything. So on matters of different suppliers in your institution, I get you. And I had just given an answer to that. Before you decide to actually procure something from a given supplier, you must, through your procurement department or otherwise, you should actually be in a position to request for samples for verification first before you adopt or you onboard that supplier. That actually makes sure that we are minimizing bringing on board suppliers that can actually not meet the supply standards that we have put in place. But that goes with first have internal established standards. Colleague, you cannot reject something when you do not have a set standard. Okay, if there is no set standard today, kindly purpose to make sure that within your operational limits there are set standards ranging from cutting across the various aspects that you bring on board. Okay, because if you have a standard, you bring in something, you do verification, you ask yourself, does it meet our internal standards? If yes, okay. If it meets the internal standard, does it meet its own manufacturer standards as declared? Okay. If it does not, then you put it aside. But if it meets internal and manufacturer assumptions as put forth, then that is something that you adopt. How do we ensure that a machine or kit is fit for purpose and fit for use? Okay. This is what I'm saying. Internal controls established. Test whatever has been put forth against internal controls. Once it meets internal controls, it means it's fit for use. But once it does not meet internal controls, it means its fitness for use is put in question. Unfortunately, fitness for purpose is actually as per the manufacturer's specifications. So you can actually indirectly also challenge the manufacturer's specifications. You have done your verification internally, it does not match what the manufacturer has put forth. So you invalidate the fitness for purpose within your localized aspect as such. Thank you.
Thank you so much. I'll bring in a question from an anonymous attendee. And the question goes, are QC material available for kits, especially H. pylori and Salmonella antigen? And then I'll bring in the second question from Martin Njogona is saying, can a facility purchase a machine for use in the lab without involving the personnel in the lab? Thank you, Martin.
So I'll start with Martin's question. Can a facility purchase without involving the personnel in the lab? It depends with your internal policies, but my suggestion is the end user must always be involved in the purchases procurement processes. Okay, unless you do not do something called supply evaluation, because if you do not involve me, at the end of the day, I find that what you brought in is not working. It means I need to do evaluation and give feedback. Do we have discussions where we give feedback in such scenarios? But my take-home point is the end user must be involved in established organizations. You find that yes, we have an external, we have a whole procurement department somewhere, but this procurement department answers to a technical personnel. That technical personnel gives guidance on what should be brought in and what should not be brought in. That is how it should actually work. Or you establish something like a procurement committee where you are part of, so that your inputs are actually felt. The only problem that we have is that sometimes some of us, we act like we are not there. Okay? the situations zenye unasema mimi bora unga bora unga. So you see, in such situations we are giving room for manipulations from other departments, because at the end of the day, when there's a wrong result from your lab, it's you who will carry the cross, my friend, whether you participated in verification or whether that actually happens in your settings or not. It's you. You see, when it comes to procurement, they will sideline you, but when it comes to shouldering the accusations that have been brought forth, it's you who will carry the day. Okay, so let us just make sure where we are not involved, let us make sure that our intentions to be involved are actually felt, are actually known. Okay, whether QC materials are available for H. pylori or otherwise, I'm aware most of the kits do not actually have QC materials for these kinds of H. pylori tests, but internally it's just good to do interlab comparisons to see how well it's functioning, lot to lot actually checks, batch to batch checks to see how it's happened. But then I'm aware of some very few companies that are actually supplying H. pylori antigen kits with QC materials. Okay, that is something that you can actually look at it. But in the event that actually the QC materials are not actually available in the, you can enroll for EQA programs. I'm aware of institutions out here that are offering EQA programs on H. pylori antigen. Okay, not to appear that like I'm marketing, but I'm aware of institutions like UCAS who are actually offering this. So you can test yourself against your peers to see actually how functionality is going on. Thank you.
Thank you. And then I'll go to Adendo Opondo. Please, if possible, can you give us any confirmed reference range of any analyte for a local population in Kenya? I have never heard of any in Kenya. And then I'll go to the second one from Wilfred, and is asking, how can labs ensure equivalence in patient care when reference ranges for diabetes diagnosis are expressed differently, some in milligrams per deciliter versus millimole per liter?
I'll start with the second question. Ranges are being expressed differently. The question is, what have they adopted as the lab? Because you cannot be expressing different clients on different reference ranges with different values in the same lab. You must, as a lab, adopt one and stick with it. Okay, that is the take-home point for him. Adopt one as the lab and stick with it. That is how it works. Okay. You cannot have a mixed expression of values or measurement units in the same lab. You must adopt one and stick to that. On matters of reference ranges, there are so many studies that have been done locally. I'm aware of studies that have been actually undertaken in MTRH just about clinical chemistry reference ranges. I'm aware of studies that are even ongoing in our research facilities, in the sense of the likes of Walter Reed. There are people there who are doing actually studies about reference ranges. Okay, so what I put forth is, what has been given to you as a manufacturer was probably done somewhere away from Africa. It was not in an African context. The testing population exhibits very different testing characteristics. But yet those reference ranges are being transmitted to you operating in Africa. So whatever that was an appropriate reference range on the other end might not actually reflect what goes on here. That is why I was saying that test it first. Test it. If it does not comply, then adjust using internal control mechanisms that must be established and declared, okay? When audits are being done, auditors always ask this question: where are your reference ranges? You show them that these are my reference ranges, okay? So how did you come up with these reference ranges? You either say, we adopted the local ones. That is number one of something that you should do, but it must be documented. Number two, we adopted the manufacturer. But if you adopted the manufacturer once, you must show beyond doubt that you actually tested those reference range on the local population for adoptability. There must be that evidence. Without that, you cannot escape a finding. So colleagues, in short, I know most of us are using manufacturer-adopted reference ranges. Manufacturer-adopted reference ranges. But then we must test on the localized population to see how it complies to those reference ranges. If it does not, there are those internal adjustment mechanisms that can actually be put forth to actually make sure that it conforms as such. Thank you.
Thank you. Is there anyone who would want to maybe ask a question or make his comments verbally? We can always allow you to allow your mic on so that you can make your comments. But meanwhile, we are very grateful. I've seen a comment from Adendo Opondo is saying, Davis, you're very truthful. The statement of Mimi Bora Unga amongst lab personnel, especially coward lab managers, is a common occurrence and is significantly affecting laboratory quality performance. Thank you, Adendo, for the comment. And is there anyone who maybe would want to comment or ask a question? I can always allow your mic on so that you can maybe raise your hand and I can allow your mic on. Otherwise, it was a very, very informative presentation. I'm sure many were wondering what this was all about, but I'm sure today we have learned something. And I think from where we sit, we have come to the very end of today's webinar. And I will take this back to our presenter, Davis, for his final remarks.
Thank you, Frederick. I will leave our esteemed colleagues with this. Remember that laboratory reliability is not just built on technical excellence. It is sustained through practical readiness. Fit for purpose ensures that our methods are scientifically sound and contextually relevant, while fit for use guarantees that those methods perform consistently in the real world. When these two principles align, we move beyond compliance. We build trust, we reduce risk, and empower better clinical decisions. Let's commit to bridging the gap between design and delivery, between theory and practice, and between potential and performance. Because in the lab, reliability isn't a luxury, it is a responsibility. Thank you, Frederick. Thank you very much.
Thank you so much for the wonderful parting shot. And for sure, I can see many of us have noted down something there in the comment section. They are appreciating the presentation. And on behalf of Lab Medicine Society of Kenya, I want to take this opportunity to thank everyone who found time to join into this conversation. I want to thank the speaker, Mr. Davis, for always finding time and always sharing what he has. And I believe that it was truly an informative session. And at this point, I will allow all of us to ask any questions that we may have. Our lines are still open. You can always drop a call on the numbers on the poster. You can always write an email on the email address on the poster, or you can always visit our website for anything that is not clear to you. Otherwise, we have come to the very end of today's presentation, and I want to thank you for finding time and joining into this webinar. And until next time, have a very fruitful